So here's our second part of the lecture on energy and metabolism. Again, keep in mind, energy is the ability to do work. All living organisms have to have energy. Nothing known to science can survive on Earth without energy. Now, how it gets the energy will vary. The efficiency of conversion varies. Does it draw the energy from organic or inorganic substances? Those are all variables and different possibilities, but bottom line is all life needs energy in some form or fashion. So the energy currency that all life uses is this molecule, the structure called ATP. So we talked about nucleic acids, and we visited this a little bit in earlier chapters and earlier lectures. But ATP, it is a ribose sugar with an adenosine base attached to it, the nitrogenous base, and then there's 3 phosphate groups attached to the ribose sugar. So I call it the little sugar house, adenosine porch, and then 3 chimneys stacked on top of each other. And as the ribose gets the phosphates attached to it, the energy level increases in what it takes to attach those phosphates. Now, to actually attach the phosphates, that requires energy. That energy comes from cellular reactions that give off energy. So those are known as exergonic reactions. So the exergonic reactions in the cell, the breaking down of energy and conversion of it, that allows ATP to form. Now you have ATP, this high energy molecule, it's all excited, it's got lots of energy because of these phosphates. Cellular processes cause a phosphate to snap off. So one phosphate breaks off, that bond is ruptured, and that releases energy. And that energy that's released goes for endergonic cellular processes, growth, development, building, repair, all those things. Now, fortunately, the ADP structure, you still have that sugar house, you have the adenosine porch, and then you have 2 phosphates, so we call ADP, adenosine diphosphate. That simply gets reassembled into ATP. Energy coming from reactions, attach the phosphate on, resetting the ATP structure. The analogy I use sometimes for this little ATP-ADP cycle is ADP is like a mousetrap. You got this mousetrap sitting in your drawer and say, oh, I need to set it. So it takes energy to pull that arm back and to set that little bar. Once it's set, it has a high energy molecule. It's got a lot of energy in it. That is equivalent of ATP. When that trap is sprung, boom, lots of energy gets released. That energy coming off is what drives growth in life and cellular processes. But again, we need an input of energy in to form ATP, and then we release energy out when it is broken down. Now, the food we eat, the digestion of that food, that is what drives the energy into this little cycle here to form ATP. So as we look at energy and pathways and metabolism, we want to look at those things we talked about in the previous lecture called catalysts. Catalysts generally come in 2 categories. Now all catalysts are going to speed up reactions, but they generally come in 2 groups. One group is called the organic catalysts. These tend to be protein-based catalysts, protein-based structures that accelerate reactions. Most of the time, they're called enzymes. In the picture here, we got this big blue enzyme that will attach to a particular substrate, and the space that they attach at is called the active site. So an organic catalyst, the organic catalyst is a protein-based substance that will accelerate a cellular reaction. It attaches to a very specific substrate, creating the enzyme substrate complex, and this causes the substrate to change into something else, moving it along a pathway, which we'll get to in just a minute here. The other type of catalysts are called inorganic catalysts. Now, these are non-living, never been alive. They're not a protein, they're not a living or a component of a living organism. A lot of times these are minerals, these are different elements. These are things, though, that will also accelerate reactions. They make the reactions go faster, they make it easier because they lower the energy of activation. Doesn't matter if you're inorganic or if you're an organic catalyst, you lower the energy of activation. So both substances will lower the energy, which makes a reaction happen much faster. Now, what we often see are these things called multi-enzyme complexes. Often they're embedded into the cell membrane, plasma membrane of the cell. So what we're looking at here is a series of enzymes that work with specific substances as they go through each step of a particular pathway. So we start out at the top with the ingredient, or the thing we call the substrate, that little circle. Now that circle attaches to enzyme 1 at the active site, and it gets changed. It gets changed into the triangle we see right here. This is called the intermediate. Now the triangle attaches to enzyme 2. Enzyme 2 changes the triangle's shape into a square. The square is now the intermediate, we would call it substrate B. That then gets moved and attaches to enzyme 3. The square gets changed into the oval, what we could call the intermediate or substrate C. That then attaches to enzyme 4. That changes its shape into our little octagon down here, or hexagon I should say, what we would call the end product. Now if you notice, it started with a substrate, had a series of intermediates, and eventually you have an end product. The enzyme just made it happen much faster. So a multi-enzyme complex is a series of enzymes that work with specific substrates to change them, change their shape, change their form, as they move through a particular pathway. Now, as we look at an example here, we have sucrose, the digestion of sucrose that follows this pattern. Sucrose is a disaccharide. It's glucose and fructose bonded together. That goes into the body. We ingest it, we eat it, we drink it. Table sugar here. Attaches to an enzyme at a specific spot. A specific enzyme will attach to this. So the substrate, sucrose, binds to the enzyme, forming the complex, and the binding causes it to break. So glucose gets released and fructose gets released. They're broken apart. The bond between them is broken by that enzyme. That enzyme then releases the glucose and fructose. Those are small enough to enter the cell, and they get converted into ATP. But that enzyme, that blue-shaped bowl thing, that enzyme literally just slides back to the beginning, picks up another piece of sucrose, goes through the pathway again. It's breaking the sucrose apart. So the nature of the enzyme is to work with a specific substrate. It will accelerate the rate of reaction. They are reusable. Quite often enzymes will work going in both directions. So if we wanted to put sucrose together, not that we can in our body, but in a plant or in certain pathways, the same enzyme will work to actually build sucrose. So lots of enzymes are functional whether you're breaking something apart or you're putting something together. Doesn't matter which pathway you're working with. They are incredibly important to maintaining the speed of any chemical process. Now, they're not making magical pathways happen. They're not creating a new pathway. They're just making existing pathways go faster and easier. And we, living organisms, have developed a a dependency on the speed of those pathways. But enzymes are very sensitive. They are sensitive to changes in temperature and in pH. Every enzyme, since they're proteins, has an optimal window of temperature and pH where it likes to work. If you go outside of that temperature range, the enzyme may stop functioning. Most of the time it completely stops functioning. So if you remember back to the chapter on food, we talked a little bit about denaturing a protein. Temperature and pH are the two things that denature a protein. So if you denature your enzyme, the pathway doesn't work. It doesn't progress. If we denatured the enzyme here and changed the shape of the active site here, turned it into maybe a square or an oval shape, then sucrose would not be digested because it can't attach to the enzyme substrate complex anymore. So the key is maintaining optimal temperature and optimal pH for the particular enzymes to function. Okay, so when we look at two enzymes that aid in digestion, pepsin and trypsin, pepsin is found in your stomach. It will assist in digestion of the nutrients when they hit the stomach because it can function in stomach acidity. But once that stomach content moves into the small intestine, pepsin gets denatured because the small intestine's pH is usually in the upper 7s to low 8s. So as pepsin gets carried into the small intestine along with all of the stomach contents, the partially digested food, it denatures. It doesn't have to become acidic to denature. It could go basic in denature. That's what's going to happen to pepsin. Fortunately for us, trypsin is sitting there in the small intestine. Trypsin takes over and continues the digestion of food and the different nutrients that are coming into the small intestine. But if by chance trypsin was ever to make its way into the stomach, we would have a valve issue or a sphincter issue, but if it did, trypsin would not work in that environment. They're very, very sensitive to changes in temperature and changes in pH. So when we look at enzymes, denaturing is the changing of the shape of the enzyme. Change the shape of the protein. pH changes it, temperature can change it. Sometimes the ionic strength of the molecules associated with the membrane or with the protein can change it, and the solubility of it can cause those things to change. The key is when you denature it, they lose their biological function. Shape equals function. Changing the shape of that protein, the enzyme, whatever protein it is, causes it not to work well. Sometimes we can renature the enzyme. We can get it back to that original shape. It depends on how badly it's been denatured. So living systems require these enzymes. They need them to be able to function optimally. Otherwise, pathways slow down and just get really, really sluggish, which for most living things, it's not a viable way to function. Okay, so as we look at enzymes, sometimes they come across this problem called inhibition, where they actually get blocked. There's two types, competitive and non-competitive. In competitive inhibition, we'll see the inhibitor actually sneaks into that active site on the enzyme and blocks the substrate's ability to get in. So the substrate can't even attach to the enzyme. It prevents it. In non-competitive inhibition, the substrate can't get in and work because the inhibitor binds to a different site called the allosteric site, and that prevents the enzyme or causes the enzyme shape to change. So the competitive inhibitor blocks the substrate. The non-competitive inhibitor basically just changes the shape of the enzyme so it becomes non-functional.